Hello and welcome to Shikara Academy's Prelims Crash Course for Science and Tech. In this particular course, for in around 20 sessions or in 20 classes, we will be trying to cover the entire science and tech syllabus that is required for your prelims examination. This course will be useful for the students or the candidates who are preparing for UPSC or the different state public service commission examinations such as TSPSC as well as APPSC. So this will act as your foundational or your crash course for science and tech prelims. Now, the topic of discussion today will be in the first class will be the cell and the genetics. So we'll start with biotechnology and bio part first, and then move on to other successive technologies like the space technology, like the nanotechnology, the defense technology, and all the other areas of the prelims syllabus. Now, this cell, we say that the cell is the fundamental unit of life. And what do we mean by when we call it the fundamental unit of life? So, we know that unicellular organisms, that is, organisms that are made up of single cell, they are capable of independent existence. So, they can survive on their own, and also they are able to perform the essential functions of life such as digestion, respiration, that is getting energy, and they are also able to reproduce, making more and more offsprings. So we can say that a unicellular organism itself, it is made up of one cell and is able to perform all the essential functions of life. Now anything less than a cell, anything less than a complete structure of cell, it does not ensure the independent living. See, independent living is the key term here. So we can also say that we can take examples of virus over here. Virus is such an entity which we cannot classify as living or dead, because virus requires its host in order to live or in order to reproduce. That is why we do not say that it is capable of independent existence or independent living. Now, the cell is the fundamental structural and functional unit of all living organisms. So we say that the virus is not entirely made up of cell. So cell is not a virus and virus is not a cell, because it is not able to independently live on its own. Looking at the history, Anton V. Leeuwenhoek was the first scientist who saw and described the cell with his simple microscope. He was a Dutch scientist and he observed the first and described the first cells, and he used a simple microscope to observe the structure of the core. Now, some years down the line, Robert Brown discovered the nucleus of the cell, and we will be discussing about the nucleus in today's class as we move forward. Now, another very important concept is the concept of cell theory. According to this theory, there are two pillars to this theory. All living organisms are composed of cells and product of cells. So all living organisms, whether it is a unicellular bacteria, whether it is a fungus, whether it is a plant, whether it is animal, whether it is humans or marine life, plants, trees, every living being, every living organism in this world is composed of or is made up of cells and the product of cells. And the second pillar is that all the cells, they arise from the pre-existing cells. So one cell give rise to the another cell. So a cell is derived from a cell only. So this is the cell theory, and these are the two fundamental pillars of the cell theory. Now we look at the structural organization of a cell. So A cell, is a fundamental unit in itself, but it has certain components or certain organization structure to it. So, a typical cell, all the cells are made up of what we call a plasma membrane and a nucleus and cytoplasm. Plasma membrane is the outer covering of the cell which separates it from the external media outside. Okay. So, if this is the cell, please note that if this is the cell and the nucleus lies in the middle, nucleus is the most dense part of the cell. This outer covering is the cell membrane, or it is also known as the plasma membrane. And the nucleus lies in the middle. It is the most dense part, and the nucleus houses the genetic matter. The nucleus houses the genetic matter, that is these DNA in the form of chromosomes. In cytoplasm is a matrix. It is a media that fills the space between the cell membrane and the nucleus, and it houses what we call the cell organelles, which are specialized parts of the cell that perform different functions. So first we look at the plasma membrane or the cell membrane. The plasma membrane or the cell membrane, it is the outermost covering of the cell that separates the content from the external environment. So the cell membrane, it separates the external environment and the internal environment of the cell. It allows or permits the entry and exit of some materials in and out of the cell, and it also prevents the movement of some other material. So it allows some material, it prevents some other material. So we say that it is selectively permeable. It is selectively allowing certain substances to enter the cell. It is selectively allowing certain substances to not enter the cell, to let it outside, and it also allows selective permeable to for the substances that are going out of the cell. So it will allow certain substances to go out of the cell or to move inside the cell. So this is also known as the selectively permeable membrane. Now, looking at the structure of the plasma membrane, one key word is the fluid mosaic model. This fluid mosaic model is associated with the structure of the plasma membrane. Okay. And what do we mean by this fluid mosaic model? That is, this cell membrane or the plasma membrane is made up of lipid bilayer. Okay. So it is made up of phospholipid bilayer and has certain glycoproteins as well as glycolipids that are attached to its lipid bilayers. Also, there are certain membrane proteins, certain integral proteins, channel proteins that give the structure, and the protein channels, they allow the passage of the substances in and out of the particular um, membrane. Okay. Now, this cell membrane, it is mainly composed of the lipids and proteins. Okay. And the major lipids are the phospholipids. So mainly it is made up of phospholipids. Phospholipids are a type of lipids and are arranged in a bilayer. So that is why we call it a lipid bilayer. And depending on the ease of extraction, the membrane proteins are be classified as integral and peripheral. So peripheral proteins, so if this is the peripheral protein, it it is situated on a periphery, and the integral proteins, they are deeply embedded, so it becomes very difficult to extract the integral proteins out of the cell membrane. The peripheral proteins lie on the surface of the membrane, while the integral proteins partially or totally are buried inside the membrane, and they are really hard to extract them. Quasi fluid nature This fluid nature of the lipid enables the layer for the lateral movement of proteins. It also enables the cell growth, the growth of the cell size, and also the formation of intercellular junction. So intercellular junction, so the junction or the attachment between the two cells is known as the intercellular junction. Secretions, endocytosis, and cell division. So this plasma membrane is involved in all the secretions, in the endocytosis and the cell division. That is when the cell divides to give up two copies of the cell. The membrane is selectively permeable to some molecules present on either side of it, either from the outside or in the inside. So it is selectively permeable. It allows only certain selective substances to enter or go out of the cell. The type of transport it being is the passive transport. That is, it does not require energy, and the active transport is the that requires the energy or specialized proteins. Now we look at the two different types of cells. We have looked at the now we are have looked at the component. We're looking at the components of the cell. That is the plasma membrane, the nucleus, as well as the cytoplasm. But before that, we have to discuss about the two different type of cells. That is the prokaryotic cells as well as the eukaryotic cells. So prokaryotic cells are small in size and are simpler cells, and they are primitive cells. We can say they are the primitive cells, and the eukaryotic cells, they are large, they are complex in nature, complex in function, they are complex in their structure. So size is approximately, the eukaryotic cell is like 100 times more greater than the prokaryotic cell. And prokaryotic cell is always found in the unicellular organisms. On the other hand, the prokaryotic cell, the eukaryotic cell, it can be found both in the unicellular as well as the multicellular organisms. The nucleus is absent. In prokaryotic cells, the nucleus is absent, and in the eukaryotic cell, the nucleus is present. So a well-defined nucleus. So in the prokaryotic cell, what happens is that the genetic matter or the genetic material, it lies inside this cytoplasm without a nuclear envelope or without a proper nucleus. But the eukaryotic cell, they have a proper nucleus that is encompassing the genetic matter, or the genetic matter is held inside the nucleus. The DNA or the genetic matter that is present is circular DNA. We also call it plasmids. And in the eukaryotic cell, the genetic matter is the linear DNA. And the chromosomes are the single haploid chromosome. In eukaryotes, we have the paired diploid chromosome. The haploid and diploid will be learning about in the later in the second half of today's class. Most importantly, the prokaryotic cell, they lack the membrane-bound organelles, and the eukaryotic cells, they have the membrane-bound organelles. They reproduce sexually as well as asexually, and the eukaryotic cells, mostly they reproduce sexually. Cell division is by binary fission. Division is by mitosis. We'll be looking at mitosis and meiosis in the second half of today's class. And the examples being the bacteria or archaic cells, examples being the plant cells, animal cells, including humans. But there are certain similarities between these prokaryotic cells as well as eukaryotic cells. Both have this plasma membrane, both have the cell membrane, both have the cytoplasm or the matrix that is housing the cell organelles. Both have the specialized structures, the specialized organelles that we call the ribosomes, which are involved in the protein synthesis. And both have DNA, but the difference is that the prokaryotic cell, they have the circular DNA, and the eukaryotic cell, they have the linear DNA. So looking at their structures, we can see that this eukaryotic cell, it is complex, it is well developed, it is more advanced as compared to the prokaryotic cell. Now the ribosomes that we were discussing, that were present in, so ribosomes, they are present in both prokaryotes as well as prokaryotic cell as well as eukaryotic cell. So what is this ribosome? The ribosomes are essential non-membrane bound cell organelles responsible for protein synthesis. In simple terms, they are the molecular machines that read the messenger RNA and use the genetic code to assemble amino acids into proteins. So here we introduce the central dogma of life. So in central dogma of life, so DNA gives rise to the RNA, and this RNA is the messenger RNA, is the mRNA, and this RNA encodes for proteins. The process, this entire cycle is known as the central dogma of life. The process by which the DNA makes copies of itself is known as DNA replication. And the process by which the DNA gets converted into mRNA or RNA is called the transcription. And the process by which the mRNA gives rise to the protein. So mRNA codes for, or mRNA produces for the proteins by attaching the amino acids to form a polypeptide chain or to give out the proteins. That process is known as translation. So these ribosomes, they are very essential for this process of translation, that is making of proteins out of the messenger RNA. Okay. Now, the ribosomes are essential non-membrane bound cell organelles responsible for protein synthesis. So, these are the ribosomes. Now we introduce the concept of plasmids. So, plasmids are the circular DNA. Plasmids are circular DNA. So, in addition to the genomic DNA, many bacteria have this small circular DNA outside their genomic DNA. So, genomic DNA is what which lies inside the nucleus, that we call the genomic DNA. And but in some bacteria, there is some small circular DNA that lies outside the genomic DNA that we call the plasmids. Now, these are small DNA are known as plasmids. The plasmids confer certain unique phenotypic characters to the bacteria. An example being uh, the antibiotic resistance that is imparted in the bacteria. The plasmids are responsible for that. And it is also used to monitor the bacterial transformation with the foreign DNA. Now, inside the cytoplasm, we have certain organelles. Organelles are the functional parts of the, or the functional parts of the cell. Organelles are certain functional part of cells, and they have each have their own specialized function. So organelles, we have the endoplasmic reticulum, we have the Golgi apparatus, we have the lysosomes, mitochondria, plastids, vacuoles, and centrioles. Please note that all these are not present in all the types of cells. So, for example, the plastids are only present in plant cells, whereas the centrioles, they are present in the animal cells. Okay. Now, this is a typical animal cell. Okay, we can see the different type of organelles, that is the Golgi apparatus, the plasma membrane, centrioles, lysosome, ribosomes, mitochondrion, rough endoplasmic reticulum, cytoplasm, nucleus, nuclear envelope, the smooth endoplasmic reticulum. The cytoplasm is a matrix that houses all these different type of organelles. Now, looking at these organelles, the endoplasmic reticulum is like a large network of membrane-bound tubes and sheets. It is of two types. We have the rough endoplasmic reticulum and we have the smooth endoplasmic reticulum. Rough endoplasmic reticulum has on their surface the ribosomes. So these granules like structure that are present on the surface or on the layer of these endoplasmic reticulum gives it a rough appearance under the microscope. That is why we call them the rough endoplasmic reticulum. RER looks rough under a microscope because it has particles called the ribosomes that we have seen in the previous slide attached to its surface. The ribosomes, which are present in all active cells, are the sites of the protein manufacture. As we have seen, the ribosomes are involved in the translation process of the central dogma and they are responsible for synthesis of proteins, that is the protein manufacture. The manufactured proteins are then sent to the various places in the cell depending on the need using the endoplasmic reticulum. So endoplasmic reticulum is a system of sheets, it's a system of tubes. So the proteins that are made, they are transported from one part of the cell to the other part of the cell via these tubes, via these sheets. Uh, that is called the endoplasmic reticulum. The smooth endoplasmic reticulum, on the other hand, helps in the manufacture of the fat molecules or lipids, and these lipids are important for the cell function. Some of these proteins and lipids help in building up of the cell membrane. So the proteins that are made by the rough endoplasmic reticulum or the ribosomes and the lipids that are made up by the smooth endoplasmic reticulum, these are very essential for the formation of the cell membrane or the plasma membrane. And this process is known as membrane biogenesis. The function, the endoplasmic reticulum serves as a channel for transport of materials, especially the proteins, between various regions of the cytoplasm or between the cytoplasm and the nucleus. The endoplasmic reticulum also functions as the cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. Example being in the liver cells. So liver is the detoxifying organ of the body. So the liver plays a crucial role in detoxifying certain toxins. So the smooth endoplasmic reticulum in the liver cells play a crucial role in detoxifying many poisons and many drugs. The Golgi apparatus. The second. So what happens with the proteins that are made by the endoplasmic reticulum or supplied by the endoplasmic reticulum? They enter the Golgi apparatus. So the Golgi apparatus is a system of membrane-bound vesicles. Vesicles are the flattened sacs. They are arranged approximately parallel to each other in stacks called the cisterns. Now, the material that is synthesized by the endoplasmic reticulum or the ER is packed and dispatched to various target sites outside, inside, as well as outside of the cell through the Golgi apparatus. So, the endoplasmic reticulum is like a factory site which has produced the protein or the substances that are essential. Now, that substance is um, now arrives into the Golgi apparatus and is now packed and dispatched to the certain locations through the certain target areas, whether the target area is inside the cell or it is outside the cell. Its functions include the storage, modification, and packaging of the products. So it kind of acts as a warehouse of these proteins or the substances that are made by the endoplasmic reticulum. And the Golgi apparatus is also involved in the formation of lysosomes. Lysosomes are another very important organelles that we'll be studying about. So lysosomes are the membrane-bound sacs filled with digestive enzymes, and these digestive enzymes are the hydrolytic enzymes. And now what these enzymes do is they are very powerful and they digest. These enzymes, they are mainly digesting enzymes. So they digest all the things that can be uh, that are not utilized by the cell. So these enzymes that are there, these are produced by the rough endoplasmic reticulum, and enzymes in itself are a type of proteins only. So enzymes are nothing but the some specialized proteins that are involved in speeding up the reactions inside the body. So foreign materials that enter the cell, such as bacteria or food, as well as some old organelles, end up in the lysosome. So if some foreign uh, material has entered the cell, such as an unwanted bacteria, unwanted other pathogen, or some unwanted food that needs to be cleared, and some organelle uh, which is not functioning properly, old and organelles, so they end up in the lysosome. What happens in lysosome? So which the lysosome break these complex substances into the simpler substances. Okay, these hydrolytic enzymes, they break down the complex substance into the simpler substance during the disturbance of cellular metabolism. For example, when the cell get damaged, if due to some reasons the cell gets damaged, so lysosomes may burst and the enzymes digest the own cell. Okay, so if by any way the cell gets damaged, or if the cell is not able to function properly, so what happens is that cell has now become redundant for the body. So the body has its own mechanism to get rid of such cells. So what happens is if the cell has become non-functional or it has become damaged, so the lysosomes, they burst. So what happens when they burst? These proteolytic enzymes, they will be scattered inside the cell, or it will ooze out of the lysosome, that is the sac, and will ultimately digest the own cell. The entire cell will be digested. That is why we call these lysosomes as the suicide bags of a cell. When a cell has to commit suicide, these lysosomes will burst, and the proteolytic enzymes, the hydrolytic enzymes that are present inside these lysosomes, or on in these sacs, that will digest the entire cell. Now we look at the some of the differences between a plant cell and an animal cell. So, firstly, there is the cell wall. A cell wall is present in a plant cell and is absent in an animal cell. Okay. Now, the animal cell, it has some a specialized organ known as centriole, which comes into picture when the cell division is taking place. The centriole is absent in the plant cell. Another important organelle is the plastid, or what we call so the plastids are the pigment carrying organelles. It has different types, that is chloroplast, leucoplast. These are different pigment containing organelles and are specially present only in the plant cell. They are absent in the animal cell. In the animal cell, we have small and several vacuoles. So, vacuoles are the cell organelles that are involved in storing storage. Okay, storage up of waste materials, also storage up of the useful materials that are synthesized by the cell. So, in the animal cell, we have small multiple vacuoles, but in the plant cell, we have a single large vacuole. Now, after the uh, learning basics about the cells, let's discuss the cell cycle. Or we know that we have learnt in the cell theory that all the existing cells have uh, come from the pre-existing cells. So all the cells come from the pre-existing cells. So the cell cycle is the sequence by which cell duplicates its genome, it synthesizes the other constituents of the cell, and eventually a cell divides and give rise to the two daughter cells. The cell divides into two daughter cells, which is termed as the cell cycle. Although the cell growth in the in terms of the cytoplasmic increase, so is a continuous process. When in the cell growth is a continuous process. Cell growth means growth in the size of cell. Growth in the size of cell, it is an ongoing process, and it is a continuous process, it is an ongoing process. But after a cell, it has like attained a certain threshold, it starts to divide. And this, the entire process is part of the cell cycle. The DNA synthesis occurs only during one specific stage of the in the cell cycle. The replicated chromosome or the DNA are then distributed to daughter nuclei by a complex series of events during the cell division. The duration of the cell cycle vary from one organism to the another organism basic depending on the cell type to cell type. So cell growth we know is an ongoing process. But in this cell growth at certain specific stage, the DNA it starts to replicate itself. Okay, so when we say the DNA is starting to replicating itself, so that means the DNA is making copies of itself. So the replicated DNA, the replicated chromosome are then distributed into the daughter nuclei. The first the nuclei divides, and so in this is the nucleus. In nucleus, we have this genetic matter that is present, this highly coiled genetic matter, and this genetic matter first it replicates and make two copies. Then this nucleus it starts to divide, which each copy of the genetic matter or the DNA being present in the each of the daughter nuclei. And the phases of this cell cycle is the first is the interphase, that is the stage between the two successive M phases. So we see the M phase as well as the interphase. So the M phase in the cell cycle, it represents the phase when the actual cell division or the mitosis occurs. So mitosis is the process by which a cell divides to form the two daughter cells, and in which the genetic matter remains constant, that is the diploid cell is giving rise to only a diploid cell. The chromosome number is constant. In a mitotic division, the chromosome number is constant. On the other hand, in meiosis, the chromosome number halves. So the M phase represents the phase when the actual cell division is happening, and the interphase represents the phase between the two successive M phases. So the M phase, it starts with the nuclear division, that is the division of nucleus. And it corresponds to the separation of the daughter chromosomes. So first, the chromosome, it has replicated itself, it has made copies of itself. So now the nucleus, it starts to divide. So nuclear division is taking place. This nuclear division is known as karyokinesis. And this ultimately ends up with the division of cytoplasm. So firstly, there is division of nucleus. Then this nucleus of the division of nucleus give rise to the division of cytoplasm, that is known as cytokinesis. The interphase, though it is called the resting phase, in which the actual mitosis process is not happening, is the time during which the cell is preparing for division by undergoing both the cell growth as well as DNA replication in an orderly manner. So firstly, we know that there is cell. This cell grows in size. Then inside the nucleus, there is DNA replication. So this entire phase is the interphase, that is in which the cell growth and the uh, replication of DNA takes place, and the genetic matter doubles inside the cell. Now, discussing the difference between mitosis and meiosis, the process of cell division. So please note that both mitosis and meiosis are the process of cell division. And the process of cell division by which most cells divide for growth is known as the mitosis. So for the growth of body, one cell is giving rise to two cells by the process of mitosis. In this process, each cell, we call the mother cell, divides to form two identical daughter cells. In mitosis, the mother cell divides to form the two identical daughter cells. The daughter cells have the same number of chromosomes as the mother cell. So if in humans we have 46 chromosomes, so after the process of mitosis, we will have two cells with 46 chromosomes each. Okay, and this mitosis helps in the growth and repair of tissues in an organism. Now we have some specific cells present in the body known as the germ cells, or cells of the reproductive organs or tissues. They that form the gametes. And these gametes, after the fertilization, they give rise to the offsprings. So the gametes, they divide by the meiosis, which involves two consecutive divisions, which results in four new cells. So if this is the mitosis and these are the chromosomes, please note that in after the mitosis, we have only two cells. We have two daughter cells, and these daughter cells, they have the same number of chromosomes. So here we have, so in this pictograph we have four chromosomes. So please note that the daughter cells also have the exact number of chromosomes, that is the four chromosomes. Now in the meiosis, it involves two successive cycles, or two successive cell divisions. And if two successive cell divisions are there, so ultimately it will give rise to four daughter cells. And these four daughter cells, they have half the number of chromosomes that are present in the original mother cell. So after in the mother cell, two daughter cells, we have this. So if the mother cell is diploid, we say that the daughter cells after the meiosis are haploid. We'll be looking at what is haploid, what is diploid in today's class, in the second half of today's class. So that was a brief uh, discussion on the cells and the cell cycle. The cell cycle also includes the cell division. Now we introduce you to the basics of genetics. Okay, so we have something called the nitrogenous bases. So nitrogenous bases, these are essentially the organic molecule containing nitrogen that are essential components of DNA and RNA. And they serve as the building blocks of the genetic information. They are categorized into two main groups based on their chemical structure: purines, adenine, guanine, and pyrimidines, that is cytosine, thymine, and uracil. So please note that we have, in this world, we have five different types of nitrogenous bases, which are classified into purines as well as pyrimidines. And they have these purines, they have it it encompasses of adenine and guanine, and the pyrimidines, they have the cytosine, uracil, and thymine. So these are the five different nitrogenous bases. Now what happens, like, when these nitrogenous bases, they are attached to a sugar, mainly the pentose sugar. When a nitrogenous base is attached to a pentose sugar or a sugar, then what we get is a nucleoside. Okay, and when this nucleoside, when we add a phosphate group to it, when we add a phosphate group to this nucleoside, then we get what we call the nucleotide. So please note that the nitrogenous base, any of the nitrogenous base, so we have 5 nitrogenous bases. So when the nitrogenous base attaches itself to a pentose sugar, then what we get is a molecule that we call the nucleoside. And when in this nucleoside we attach the phosphate group or the phosphate molecule, then what we get is the nucleotide. And this nucleotide is the basic unit that forms the DNA. So we see we have a nucleotide. This chain of nucleotides gives us the nucleic acids. So we have a nitrogenous base. On nitrogenous base plus sugar gives us the nucleoside. Nucleoside plus the phosphate group or the phosphate gives us the nucleotide, and the chain of nucleotides gives us the nucleic acid, is known as the nucleic acid. So basically we can say that nucleic acid is made up of nitrogenous base plus pentose sugar as well as the phosphate, because a nucleotide is made up of these three things only. So we can say the nucleic acid is also made up of the nitrogenous base, the pentose sugar, and the phosphate group. Nucleic acids are of two types. We have the DNA, deoxyribonucleic acid, and we have the RNA. DNA, RNA. DNA is the deoxyribonucleic acid, and RNA is the ribonucleic acid. Now, what is the difference between a deoxyribonucleic acid or the ribonucleic acid or the RNA? So, this DNA, both DNA and RNA are made up of 4 types of nucleotides or the 4 types of nitrogenous bases. We have adenine, thymine, guanine, and cytosine. In DNA, we have the 4 nitrogenous bases, that is ATGC, adenine, thymine, guanine, cytosine. RNA, on the other hand, it has adenine, uracil, guanine, and cytosine. So only the difference is in DNA we have thymine, in RNA we have this uracil. So this is the difference when it comes to the nitrogenous bases. When it comes to the sugar, so the sugar here is the deoxyribose sugar, and in RNA the sugar is the ribose sugar. Also, looking at the strands, a DNA, a stable DNA, a stable structure or stable molecule of DNA will be double stranded, and RNA predominantly they are single stranded. Looking at the bonding between these two, what we call the nitrogenous bases. So a adenine molecule is joined to the thymine by 2 hydrogen bonds, and a guanine molecule, the the guanine nitrogenous base is attached to cytosine using the 3 hydrogen bases. So this is known as the base pairing. The nitrogenous bases are paired with each other. Adenine pairs with the thymine in DNA with 2 hydrogen bonds. The guanine, it with 3 hydrogen bonds, it binds with the cytosine. Similarly, adenine binds with uracil using 2 hydrogen bonds. and the guanine, as in the DNA, binds with the cytosine using the 3 hydrogen bonds. Okay. So now we have looked at the what a DNA is actually made up of. So DNA is a nucleic acid, and a nucleic acid is made up of a chain of nucleotides, and nucleotides are made up of nitrogenous bases, pentose sugar, and the phosphate. Now, what is a chromosome? So now, if this is a DNA, and we know that DNA, it occurs in double strands. So if this is a DNA, so it is coiled inside the nucleus. So DNA is housed inside the nucleus and is coiled. So this coiled form of DNA is known as, or this condensed form of DNA is known as chromatin. So if this is our DNA, and this condensed form of, or this highly coiled form of DNA is known as chromatin. That is the condensed DNA. This condensed DNA, when we further and further we condense this, further and further condensation of this DNA molecule and packing of this DNA molecules gives us the chromatid, which is the maximum condensed DNA. This is the maximum condensed DNA is the chromatid. Now, these chromatids, when they are present in pairs, so the pair of chromatids gives us the, is known as the chromosome. So if this is a chromatid, so chromosome will be like a pair of these chromatids, and this will be the chromosome. Clear? DNA is a linear molecule, and that DNA is a very long molecule. It needs to be condensed. It needs to be highly coiled. It needs to be condensed in order to fit inside a tiny cell, and even though inside the tiny nucleus. So what we do is, it is present in a very condensed form. So the condensed form of DNA is known as the chromatin. And when this chromatin achieves the maximum condensation possible, so maximum condensed form of DNA is known as the chromatid. The chromatid, a pair of chromatid, is called the chromosomes. So now I am hopeful that you have the difference between, you have got the difference, the difference between the DNA and the chromosome is now clear. So in human body, we have 23 pairs of chromosomes. Please note that these are not the pair of chromatids, but I am writing these 23 pairs of chromosomes. That means 46 chromosomes are present in the human body. When we say human body, that is inside a human cell. We have 46 chromosomes. Out of these 46 or the 23 pairs, the first 22 pairs, are known as autosomes, and the 23rd pair is known as the sex chromosomes. So first 22 pairs, so we have in total we have 23 pairs of chromosomes. So first 22 pairs are known as autosomes, and they control the characters that are common in both males and females. Human body, we have 23 pairs of chromosomes. Out of them, the first 22 pairs are called the autosomes, and they control the characters, the traits that are common to both men and women. That is their like the features or the characters that are common to both males and females. The 23rd pair is the sex chromosome. Now, the sex chromosomes is of two types. We have the X chromosome and we have the Y chromosome. The X chromosome is known as the female sex chromosome and the Y chromosome is known as the male sex chromosome. And as you have guessed, the female sex chromosome, it controls and it imparts the characters that are specific to females. Also, the Y chromosome imparts the or controls the characters that are specific to males. And when in size, this X chromosomes is larger in size and it is smaller in size. Controls female characters. This Y chromosome controls the male characters. Now, we have come across a term that is haploid diploid. So, what is a ploid? So, ploid is the one set of all the chromosomes in a cell. We have two set of chromosomes. So we say we have a pair of chromosomes. We have 23 pairs. But when we take a set-wise thing, so we can say that we have the N in humans is 23 because we are taking only one set. When we take the two sets, we say that our cells are diploid. That is, we have 46 chromosomes. That is, in two sets, we have 46. So here, if N, that is the ploid number, is 23, then we say it is monoploid. That is, only one set of the chromosomes is present. For 2n, we have 46 chromosomes and we call them diploid. That is, the two sets of chromosomes are present. Similarly, we have 3n, 4n, 5n, and these are commonly called the polyploids. We have monoploid, diploid, and we have polyploids. So, humans, we see in humans, we have only diploid, and in the gametes, we have monoploids. Other types of ploidies, the higher polyploidy is not observed in humans. Now, what is haploid? So, haploid means half the number of ploids. And when we say half the number of ploids, that means, so here we have a diploid that is having two ploids. So, what will be the haploid of a diploid? So, we can find it. Haploid means half the number of ploid. When we say half the number of ploid of a diploid is 2n. So, we can say the haploid of a diploid is n. That is, we know it is monoploid. Okay. Now, what will be the haploid of a tetraploid? So, tetraploid means 4n. So, haploid means we are making it half. So, it will be 2n, that is diploid. When we were discussing about the mitosis, we say that the mother cell is diploid and the daughter cells are diploid. Okay. So, we say that when the gamete formation takes place, we say that gametes they are haploid because they have undergone the process of meiosis. So haploid means that they have the half the number of ploidy. Okay, half the number of ploids, that is they have 23 chromosomes, and the diploid cells, they have the 46 chromosomes when we talk about the humans. So that's all for today's discussion. In the subsequent classes, in the subsequent live classes, we'll be taking the our discussion forward on this basics of genetics, and then we'll start with the biotechnology. Thank you.